Yes, good. I'm not Paul Dennis, as you can see. It's not deep fake. I'm Yuri Sabin, and I will be replacing Paul Dennis today on this webinar. And it's great to see the crowd of experts coming in. Experts in specifically deconvolution, protein work, top-down, intact mass measurements. It's great to see you guys here. I'm helping some of newly arriving people to get to the participant list here, and I will be monitoring that. But I think we can get started, and my pleasure will be now to introduce our speaker of today and to say a few words about the Proteoform Thursdays, although I'm pretty sure that everyone on this call already know what it is and why we are here. So here's Inge coming. Good. So really a good crowd coming in. So please, Kyowon, can you tell me that you see well the first slide? Yes?
Yes.
Good.
Yeah.
Okay.
Yeah.
So as it is stated here, it's a pleasure to welcome you to the Proteoform Thursday on March 17th. And our speaker today is Dr. Kyowon Jeong, who is currently a scientist at the University of Tübingen in Germany. The title of the talk is shown here, and it tells us that, yes, it will be about deconvolution. Yes, it will be about protein analysis and intact mass and all the other measurements that we are interested in. Before Dr. Jeong will start, let me just tell a few words about the Consortium for Top-Down Proteomics and why we are doing what we are doing and how we are trying to make an impact with these Proteoform Thursday webinars. The consortium itself has two main objectives. One is to build a community and to guide the technology. And we talk about technology as real, the top-down mass spectrometry, the top-down proteomics. And as a means to build the community, we provide education, training, and organizing these type of workshops. And as a guide in the technology, we try to help people to develop protocols, or sometimes we take the lead and make inter-laboratory studies together and give the initiatives, make the initiatives jointly with the community. And a few of them I will go through in the next slides. So our ongoing initiatives are listed here. One of them is native top-down projects to provide a guidance on specifically the native ionization, the native handling of complexes and proteins for top-down, and elucidate how this affects the high-order structure proteins. The next initiative we are currently continuing is the next step in our introduction of the proforma notation, which we introduced already a few years ago. And this time we are doing it in collaboration with HUPOR on that is working on these similar topics. The next initiative is capillary electrophoresis mass spectrometry, and to understand what impact CE can make on top-down, how useful it can be, and what could be the best practices for the successful application of CE in top-down. So that's a very interesting initiative that is already ongoing. And last but not least, the Human Proteoform Atlas. So that is the infrastructure for all of us that would host the proteoforms, okay? Already we're going from proteins, protein families, peptides, to proteoforms, and that's the key to the Human Proteoform Atlas. It's a very hard work to do, but there are many people involved, and that is progressing pretty nicely. That's, again, an international effort with people from many countries around the world contributing to that. Recently, we, as a consortium, we published a paper in Science Advances called the Human Proteoform Project. And this paper is an open access, and I encourage you to read it if you haven't done so. I'm sure that many of you have already found this paper and read about it, because it's really a very interesting read, and it provides our vision and considerations for the future projects. And if you will be interested to join these efforts, it will be we will welcome you very much to do so. Now, coming back to the topic of today's meeting here, the deconvolution. And before we conduct all of our Proteoform Thursday webinars, we ask you a question. So what is the most challenging part for you in this specific topic? And not surprisingly, the deconvolution is the leader in the charts for this webinar. Well, that's why we are here, right? So deconvolution was told as or was shown as one of the major limitations and challenges in treating the top-down data. Database search and quantitation, data acquisition, these were the minor components compared to deconvolution. So let's see if it's true, and then let's see if Kyowon can really help us out there. What were the other concerns on data handling specifically? They are listed here. These are the concerns that people who made the answered the charts, who replied to these questions. And as you can see here, again, that is about the deconvolution, how we can update the deconvolution on the fly, how we can do the FDR control, how we can do this and that. So it's really a very good selection of concerns. So you can read through those. And you can see that also people are worried about reproducibility in fractionation and the sample preprocessing, scoring, and so on and so forth, as well as going to the more practical questions. How do you set up top-down workflows in a new lab, or when a new lab is starting to use a top-down? So all these questions are also why we are here today, and what we are going to discuss in the presentation and afterwards. So very good feedback. Thank you very much for providing these details. And as final words about the Consortium for Top-Down Proteomics, very welcome to join the community if you haven't done so, and very welcome to become a member and help us work together on making top-down better and more widely spread and with increased impact. So welcome to join. And we also very much thank the financial support of our sponsors, Thermo Fisher Scientific, Bruker, Sykes, Agilent, and Pfizer. And if you see that your company would like to help us further, very welcome. Please contact Paul Dennis directly, the real Paul Dennis, not me. And you can see that you can provide support on different levels. And based on that, there will be the benefits from this support. But this will be very much appreciated, because we are a non-for-profit organization. So finally, we get to the main information from this presentation, that we're going to start the seminar. And again, the seminar will be given by Dr. Kyowon Jeong. And Dr. Jeong, he did his PhD in the group of a very famous Pavel Pevsner. And we know that Pavel and his group made really very important contribution to top-down proteomics, to top-down mass spectrometry, and not only mass spectrometry, also other areas where mathematics and programming and the algorithms that they were developing were really making a very important impact. But Kyowon did not do top-down at that time, as he told me. So during his PhD, he was not involved in these projects. But as time progressed, he realized that top-down is the best method, is the king. So he switched sides from bottom-up to top-down, and now he is doing top-down full-time in the University of Tübingen in Germany. And he's going to tell us what is the current status in the deconvolution, and specifically on the flash suite for the analysis of top-down proteomics data. So thank you very much for coming, and please leave your comments in the chat, or we discuss them later. And the floor is yours, Kyowon. Please, we are looking forward to your presentation.
Okay, thank you very much for your kind introduction, and I will share my screen. This one. Okay. Now you see.
Yes, it's good.
Okay. So this is the full screen, right? Okay, perfect.
Yes, good.
Okay. Okay, thank you very much. So hello everyone. I am Kyowon Jeong, a postdoc in Kolbach Lab in University of Tübingen in Germany. Before I begin, I want to say that I'm really thrilled to have such a wonderful chance to present my project in CTDP Proteoform Thursday talk, and would like to thank to the organizers and all the CTDP directors. Today, I want to talk about our recently developed softwares for the analysis of top-down proteomics data, collectively called the FLE suite. And I think I could cover a few topics that you just introduced. And I will also introduce ongoing projects and would like to share a few points that I think are important for top-down data analysis. Okay. For us bioinformaticians, the major hurdle in top-down data analysis is the complex signal structure of proteoform ions. Let's begin with peptide ion signals. And due to relatively small masses, peptide ions often have small charge values. And often they usually come with small numbers of isotopes. So simple. Not that simple, but anyway. But in case of proteoforms, they generally have wide charge ranges and so many isotopes. So a single kind of proteoform ion results in more than 100 peaks in the spectrum. This illustration shows only 3 charge states, but we often observed more than tens of charges from a single kind of proteoform. Then how this distinctive nature of proteoform ion complicates data analysis? Assume the LC features of the same color here represent the features from the same proteoforms. They often coelude and have very close MZ values each other. And in the real spectra, there is certainly no color, and it is very hard to distinguish which features are from which analytes. Okay, I should admit. Okay. And when we see a specific MS1 full scan, the peaks are often very dense and redundant. And in MS2 spectrum from these highly charged precursors, there are 2 very different populations, one with very highly charged and the other with very lowly charged fragment ions. And this heterogeneity does not help us in any way. These complexities in different levels complicate various proteoform analysis, including identification and quantitation. And to disentangle this complexity, mass deconvolution is often used. By applying mass deconvolution, we go from complex ion peaks to a simple list of ion masses, as in this right side. In deconvolved mass dimension, data analysis becomes far simpler than in raw MZ dimension. And for sure, far simpler does not necessarily mean that it is actually easy. We still need top-down MS-tailored softwares for proteoform identification, quantification, and even data acquisition for the best results. Many excellent computational tools for top-down proteomics have been introduced in the last decades. And this list only shows a few of many such tools. They all come with their own strengths and specific applications, and also certainly with some limits. And in recent years, we developed Flash series software tools for deconvolution, data acquisition, and proteoform quantification within our OpenMS software platform. The first software we introduced was Flash Deconv, an ultra-fast feature and mass deconvolution tool. It is based on a simple pre-processing of spectra to accelerate the charging step, and it showed not only very fast, but also accurate deconvolution results. This work was done with Ji-Hyung in our lab and with Manzi and others in an Schulete lab in University of Hamburg. And the speed of Flash Deconv is mostly done in the charging step. For decharging, Flash Deconv tries to find peaks from the same mass with different charges. They make a specific pattern in here I call charge patterns. In the original MG space, the difference between consecutive charges Z and Z+1 is given by this formula in the bottom side, which depends on the mass value, denoted as M2 here. But here I found that if we take log for MZ values, the distance between them is given by the formula in the right side, and suddenly the mass term vanishes from the formula. This means that the charge pattern is fixed for different mass values. So by taking log to all peak MZ values and then scanning this universal pattern through it, we can quickly find the charge patterns and perform discharging very fast. Thanks to this simple trick, FlexConf showed orders of magnitude faster runtime than other commercial softwares like Extract or Respect. The left side compared the runtimes from different deconvolution tools for complex mouse sample. FlexDeconv, here FD, took only 7 minutes to analyze the whole data set. And for both MS1 and MS2, FlexDeconv typically achieves less than 20 millisecond processing time per spectrum. And also FlexDeconv output far fewer mass artifacts than others, as shown in the right side graph. The blue bars represent genuine masses, and other bars represent masses interpreted as artifacts. And we released FlexDeconv 2.0 beta plus version actually a few days ago with lots of new features such as GUI, various output formats to interface with other tools like Topic and MS Pathfinder T. The most recent version with instruction is available from OpenMS homepage, given in this webpage. And by leveraging this millisecond order of runtime from FlexDeconv, we also implemented data acquisition method dedicated to top-down proteomics called FlashIDA. FlashIDA run inside of the thermal machines. The MS1 spectrum is provided by thermal eye API real-time to FlashDeconv that performs real-time deconvolution. Then the deconvolved spectrum is further processed and converted to mass quality spectrum, from which high quality masses are selected. The selected masses are converted back to MZ value and fed back to machine again through IAPI. FlashIDA avoids correlated precursors and controls isolation window size real time to select high quality precursors. FlashIDA was developed with Masha and Vladimir in Jensen Lab in Southern Denmark University. With this relatively simple workflow, we were able to make a large boost in terms of number of proteoform-level identifications. We compared FlexDeconv runs, FI datasets, against standard DDA runs for intact proteins, ST datasets from E. coli lysate single runs. The latter digits 30 and 90 specify the retention time gradients. In this bar graph, bluish bars are from FlashIDA and reddish bars are from standard DDA acquisitions. FlashIDA resulted in almost twice proteoforms from the same retention time gradient runs, or alternatively, it achieved a similar proteoform count to standard acquisition with only about a third of instrument time. And FlexDeconv Q is the next software we developed. It is a feature-level label-free quantification algorithm for top-down proteomics developed based on FlexDeconv. FlexDeconv Q is mainly developed by Ji-Hyung Kim, a PhD student in our lab, and it has also quite a fast processing time. Runtime is comparable with FlexDeconv, and in the feature level, it resolves correlating signals. It consists of 4 steps. It starts with detection of individual ion chromatograms from peaks in centroided MS1 raw spectra. And using FlexDeconv, detected chromatograms are deconvolved and grouped per mass. Before calculating the quantities of each mass to get accurate quantification, correlating signals are resolved. So assume there are 2 proteoform signals, green and orange. The red LCMS feature is the overlapping the feature. FlexDeconv Q takes the non-overlapping features and from them generate theoretical shapes. And with these theoretical shapes, the overlapping red feature is reconstructed by solving a non-negative least square problem. This shows an actual observed signal from E. coli lysate dataset. Green and orange features are from different proteoforms, and red one is overlapping. And the right side shows how FlexConv Q resolved the coelution. And this resolving step gives an accurate quantity ratio as well. To benchmark FlexConv Q, we took E. coli lysate technical triplicate. We tested FlexConv Q against FlexDeconv, Respect, and ITOP-Q. This table shows the number of quantified proteoforms from each replicate and also the number of jointly detected proteoforms. FlexConv Q found the most jointly detected ones and also showed high portion of overlap. And we drew the mass distributions of jointly detected proteoforms. Red FlexConv Q and green Respect showed relatively wide mass ranges, and normal FlexConv Q and ITOP-Q, normal FlexDeconv and ITOP-Q proteoforms are gathered in small mass ranges. And most importantly, we looked at the quantification accuracy. The green boxes show the coefficient of variation or CV values of low abundance, and orange boxes are the CV values for high abundance masses. For both cases, FlexDeconv Q showed minimum CV values, showing the FlexDeconv Q is accurate and also works well for low abundance proteoforms. But still, we have relatively high CV values exceeding 0.2 in many times, meaning that we might have room for more improvement. So far, I presented Flash Suite softwares, and I hope you were able to see the big pictures of what's going on with Flash Suite softwares. And in the latter part of this talk, I would like to introduce some of our ongoing projects. Some of them are almost finished, but others are in early stages. But I would like to take this chance to share the progress and hope some of the points that I found during this progress are beneficial for audiences. First of all, I want to show you a few example applications of FlexDeconv that have been done with Professor Dave Tapp. Dave and I have been preparing a manuscript about comparative study on top-down proteomics deconvolution and identification tools. Out of all great contents, I would like to present this part about comparison between deconvolution tools. This figure by Dave shows the overall deconvolution analysis pipeline. We use both Bruker QTOP and thermal Orbitrap datasets. And for deconvolution, we use Bruker vendor software Snap2 and MaxEnt. And for sure, we use FlexDeconv combined with Spectra Merger that I will explain later. And TopFD in Top-Pick Suite, Mascot Distiller, and lastly, Extract have been used. All different outputs from different tools have been converted into MS-Align format, and then they were identified by topic at 1% proteoform-level FDR. This bar graph shows the numbers of deconvolved MS2 spectra, the numbers of proteoform spectrum matches, and numbers of proteoforms from left to right. This is from human QTOP dataset from Dr. Kel Brown's work in Inga group. Different colors represent different fractions. FlexDeconv reported a relatively small number of MS2 spectra, but the identification rate from them was higher than others and reported the most proteoforms. One of the reasons why FlexDeconv reports less deconvolved MS2 spectra is that it uses rather harsh filtration of precursor masses. For example, it does not select MS2 that may contain multiple proteoforms, in other words, chimera MS2 spectra. The reason of this harsh filtration will become clearer in the later part of the talk. Bruker software also reported not so many deconvolved MS2 spectra. This is because multiple MS2 spectra within a fixed retention time window were merged into a single one. Extract also performs similar MS2 spectra averaging, but in the case of this averaging is done for each MS2, so the total number of MS2 spectra does not decrease. I found that this merging and averaging is very beneficial, in particular for QTOP dataset. So FlexConv now uses Spectra Merger tool, from OpenMS to perform this averaging technique that is similar to extract. And this merging can be done easily by setting up a parameter in the new version. For this data set, FlashDeconv and Bruker software were comparable to each other. We have a few additional queued-up data sets, and the tendency was similar. But I would like to emphasize FlashDeconv reduces the processing time dramatically. One interesting observation from this result is that the performance gap is quite large. This gap solely stems from the deconvolution method because others are kept the same. This shows the importance of deconvolution for the proteoform identification. So for QTOF, FlashDeconv seems to generate quite nice results, and we also evaluated it in Orbitrap dataset from E. coli lysate. For Orbitrap, extract reported the most proteoforms. TabFD also generated many. The difference already starts with the number of deconvolved MS2. Maybe FlashDeconv is filtering too many precursors, or others are allowing too many precursors. We'll see. And in the case of mascot distiller, the result was significantly compromised than others. And we are still checking what is the main cause of this low performance for Orbitrap dataset. Again, this large difference was observed between different deconvolution methods. Most obvious messages from this above analysis was different deconvolution generates different proteoform count. Deeper analysis will be provided in the upcoming paper by Professor Tapp, but here I will give a simple analysis on overlap between different tools to see if difference is only for proteoform count or for other aspects. Okay. I examined overlaps in two different levels. First, protein overlap between two runs is simply the protein oxidation overlap. Second, proteoform level overlap is a bit more specific. Two proteoforms overlap if their proteins overlap, and their amino acid sequences are the same, and their nominal masses are the same. I even did not include retention time similarity for this measure. The nominal mass overlap can be thought of as the MS2 precursor nominal mass overlap. And the second, amino acid sequence overlap does not affect the total overlap count much, as it is quite rare to have different sequences when two proteoform masses are the same. So the most important two factors are the protein oxidations and the MS2 precursor masses. The evaluation metric was simply the overlap coefficient. From this Venn diagram, the overlap coefficient is the number of intersecting elements divided by the smaller set size of the two. When it is 0%, no overlap is observed. And when it is 100%, then we have a perfect overlap. We took two replicates from the above Orbitrap E. coli samples. And for the first replicate, we used FlashDeconv, and for the second, extract for the deconvolution. For both, topic was used for ID with 1% FDR threshold using popular target decoy approach. When two results were compared, we observed 85% protein overlap coefficient. Not very bad, I would say. But in proteoform level, it was only 46%. This is very low reproducibility, but maybe the sample is to blame, maybe even if they were already published and examined by the authors. So to see who is to blame, I removed the sample variance. Let's analyze exactly the same sample using just two different deconvolution methods. Okay, in protein level, we have 92%. Well, not so bad again. And for proteoform level, oh, this is very low again, 51%. Okay, I do not blame the samples anymore. Well, but maybe this is the case in which FlashDeconv and extract are complementary each other, maybe a win-win game. Then again, I removed this possibility by only taking the proteoforms from the same MS2. They should be the same because they are from the exactly same MS2 spectra. In protein level, the overlap was 96%, still a bit too low. I wish I had like 99%. FDR is 1%, so it should be at least 98%. Then how about proteoform level? It is only 49%. even lower than the previous one. This explicitly shows the precursor masses from two methods do not match. It is very serious, either for flash decom for extract or for both. But what is even worse is that this shows that proteoform level FDR estimation is failing so bad. And this is the main reason why flash decom filters out low quality precursors. Okay. And this different level of FDR is really nicely described in this paper by Leduc et al. And our simple analysis shows that how important to distinguish between different levels of FDR in top-down studies. Okay, now I took only flash decom and designed the following analysis. For this typical deconvolution ID pipeline, I added a devil block in which arbitrary masses between 10 and 20 are added to or subtracted from the deconvolved precursor masses. For example, when the reported precursor masses are like this, some random masses added or subtracted and just ID by topic. For this search, the number of proteoform spectrum matches reduced only by 10%, not by 99%. And even worse, the number of proteoforms increased by 200%. This is expected because all different random false precursor masses will be interpreted as novel proteoforms. So one can guess that most false positives in top-down searches are from precursor mass errors. This gets serious, especially when blind modifications are allowed, like topic, and when many modifications are allowed per proteoform identification, the same problem occurs. Then why our great target decoy database cannot control these sorts of errors? A simple way to see if decoy hit works is to compare score distributions of decoy hits and actual false positives. To obtain false positives, we searched the E. coli dataset against human protein database. This is FP1 set. And we took the false positives with the incorrect precursor masses from the previous devil block pipeline. This is FP2. And lastly, we have decoy hits from decoy-only database search. Now this shows the match score distribution of decoy hits. The score, the higher, the better. And the second shows the distribution from FP1. They are quite similar, so decoy hits well represent false positives in FP1. And this is the distribution from FP2. If you compare the x-axis scale, we can easily see decoy and FP2 have very different distribution. Not only this shows decoy cannot simulate false positives from precursor mass error, but also indicates that score cutoff that do not take precursor deconvolution quality into account would have the same issue. Then what is the solution? We are trying the approach similar to condenser, which used the notion of decoy MS1 features to control MS1 feature match error rate. By using decoy MS1 features with percolator, they showed less than 5% feature level FDR. Likewise, we could extract features from precursor deconvolution and use decoy precursor masses with percolator. This is an ongoing project with Ayesha Ferrotz, a new PhD student in our group. So far, we thought about how to take precursor matter errors into account. Another way to resolve this FDR issue is to just remove all precursor errors. Maybe this is too dreamy, but we are trying to improve the deconvolution quality using deep learning to reduce and hopefully remove deconvolution errors. Deep learning is getting lots of popularity, and we already observed many success stories in our Proteomics society as well. I'm only a consumer of these techniques, but luckily I have my brother, Professor Kyowon Jeong, a real expert in deep learning, and his student, Yerin, working for this project together. From our lab, a master student, Christian Pellant, is leading this project. First off, let me give a little bit of background on two different deep learning techniques. The first one I want to show you is recurrent neural network, or RNN. RNN is used for really various problems, but I think introduce this text classification problem. where we want to classify a sequence of words into good or bad. This is a good comment, right? And the second one is a bad comment. And the third one looks bad at first, but turns out to be very good. So this classification has variable input sequence length and relation between tokens, in this case words, should be considered for successful classification. And RNN is known to work very well with such problems. And second popular architecture is convolutional neural network, or CNN. And CNN is often used for image classification. For example, we want to classify the figures into cat or dog classes. The issue in this image processing is the target's image size varies and direction or location of target also varies. And CNN is known to work excellently for this kind of problem. Then what is the connection between convolution and RNN or CNN? Let's revisit the signals from this proteoform. These peaks of different charges will be moved to the 3D space in left side with mass, charge, and intensity axis. The first blue peak can be moved like this. The first peak comes to here, and the others come one by one. And the next charged peaks appear in the next charge axis, and the purple peaks appear in the next charge. Now we encode these peaks into a sequence of tokens. The blue peaks form the first token, like this, and the second green peaks make the second token, the last purple, the third token. Note that the tokens contain similar signal shapes. When the signal changes, for example, their isotope pattern changes and have more charge states, we have more tokens with different signal shapes. For very small masses, the sequence length will become very short. Lastly, for noise, we usually have a very low relation between tokens. So overall, the input have variable lengths and signal has high relation between tokens and noise has low relation. So RNN might be the best for these inputs. And the second method to see the signal is simply to see it from above. Then the signal looks like this. It has a checkerboard pattern. If the signal changes, we have differently sized checkerboard. For small molecule, we have a very small checkerboard. Lastly, noise make rather scattered dots. So overall, target image size varies. Signal and noise have different patterns. So CNN looks a good method for this problem. CNN is already used for deconvolution in topic suite as well, with the name ENV CNN scoring. For training, validation, and test, we prepared half simulated datasets. We have 3 classes instead of 2 actually. Correct class masses are obtained by collecting deconvolved MS1 masses of very high signal to noise ratio. And noise masses are obtained by shifting the signals in correct masses by 10 Thompsons. Lastly, we also have mass artifact class that represents charge assignment errors. This is the RNN result. We used 3 different RNN models: simple RNN, LSTM, and GRU. LSTM and GRU are more advanced RNN structure. When you ran 3 times with different sets, all showed very high accuracies, exceeding 0.95. And LSTM showed the best performance, and GRU followed the next. One interesting observation is that RNN works much better for larger masses. This makes sense because larger masses often have longer input lengths, and RNN can effectively exploit the information in long input sequences. Then we tested with simple CNN model, different from RNN. We should fix the maximum range of charges. We tested with 10 and 50. The maximum isotope length was fixed to 102. The results from CNN were actually better than RNN, and the average accuracy was almost 0.97 when 50 charges were used. CNN also worked better with more charge states, but the difference between narrow and wide charge ranges were not very high. Here we show a few real examples of correctly classified masses. They are all precursor masses deconvolved by topic and identified by topic, deconvolved by top FD and identified topic, sorry. Blue bars are correctly positioned peaks and red are incorrectly positioned peaks. Peaks are binned and intensities within each bin are summed. The blue bars actually form nice isotope pattern shapes for this correct class. And they are the noise class masses. We see lots of red peaks and blue peaks do not form good shapes. Note that they are still identified precursor masses at 1% FDR. And this is another noise class peaks. This looks a very perfect shape, so looks like a misclassification. But we found that this is actually mislocated peaks by 2 Daltons. So we observed that deep learning can even find small monoisotopic mass errors. And lastly, we revisit this overlap analysis. The proteoform-level overlap coefficient for the same MS2 was only 49%. We post-processed these precursors using our deep learning filter. Then we had 85% proteoform overlap. This is very nice, but actually we also lost 28% of overlapping proteoforms. Maybe it is the case that common mass artifacts are removed together because different from noise, mass artifacts are reproduced. We still need to check if this is actually the case. But when considering 75% of extract and 55% from Fleischdecomp were filtered out, this result looked not so shabby. So in summary, I introduced Fleisch suite software, including deconvolution, acquisition, and quantification tools. And from comparative studies, we found that Fleischdecomp performs well with QTOP datasets. And I pointed out precursor deconvolution error may be the main cause of proteoform-level FDR underestimation. And lastly, I showed deep learning could be helpful for mass deconvolution error filtration. A few future directions. We are also working on analysis of native MS datasets. We are closely working with Dr. Kyowon Jeong and Professor Lu, and also with Tanya Habeck and Professor Lemit. And we collaborate with Tolle Lab for different modes of Flash IDA. Philip and Conrad are implementing Flash IDA global targeting mode with us, which will be presented in ASMS this year. And with Dr. Petras, a new PI in our university, we will try to de novo sequence snake genome proteoforms. We are also planning to implement a new identification tool within Fleisch suite, which will fully use all the information from Fleischdecomp side and Flash IDA side and equipped with the proper FDR control. And after all, in the long term, Fleisch suite will be released that combines all the above tools and features as a part of OpenMS software. With that, I would like to thank to my colleagues in Kolbacher Lab, Jihyung, Ayesha, Christine, and also Dr. Timon Zaxenberg. He helped us with OpenMS development and with lots of discussion and sharp insights. And I deeply thank to my great boss, Professor Oliver Kolbacher. He always shows astonishing leadership. I thank to Manzi, Seti, Laura, and Professor Hartmut Schletter for great contribution to Fleischdecomp project. Masha, Vladimir, and Professor Ole Jensen in Jensen Lab together developed Fleisch IDA discovery mode. I really appreciate their great inputs and critical discussions. I hope we can finish the revision safely and quickly. And I'm grateful to Philip, Conrad, and Professor Anders Tolle. I believe we can make excellent results soon. And one of my best colleagues and my friend, Professor Dave Tapp, now visiting Champotruc Lab in France. I'm very thankful that I met you in Twitter and believe we will have lots of fun projects together. And of course, I thank to my deep learning consultants, my brother, Professor Kyowon Jeong, and his excellent student, Yerin, for all the great inputs. And lastly, I thank to all the funding resources and deeply thank to the audience for your attention. Thank you very much, and I will gladly take questions.
Thank you very much, Kyowon. You can hear all the applause appearing on the screen. As you can see, there are many people from your acknowledgements are on the screen. That shows, that really it's a community, already a community around the Flash suite, and it's pretty obvious why, because of the interesting results that you have shown to us. And it's also great that it's a resource that many people will be able to use. So congratulations to that. And thank you to you and to Professor Oliver Kolbacher for all these initiatives.
Thank you very much.
The floor is open for questions. I see in the chat we have comments like, yeah, that was great, fantastic, everything good. So, and there is a question coming from Lisa Anderson from MagLab.
Okay,
so
great.
Does flash deconvolve all the spectra one by one, or can you perform post-FT spectral averaging sliding window deconvolution as well?
Both. So a default mode is one-by-one deconvolution. It is already very fast, but also you can perform like merging all the MS2 spectra into one, or MS1 spectra into one single spectrum, or you can do the averaging sliding window thing. And there is a option. I forgot the name. It's like merge method. You can find it from the homepage. You can just set up the option, then you can select the way of deconvolution.
Thank you. And the sliding window deconvolution is already implemented, right?
Yes.
It's an option, right?
Yeah, it's an option. Yeah. Okay. Yeah, and also I found like merging all the spectra into a single one is really effective, especially for native math. Like Unidec was doing that already. So I also kind of mimicked, their approach.
So how do you merge? At what stage? Is it profile data or centroid?
It's centroid. Yeah, I tried both actually, but I didn't see lots of difference between profile and centroid. So I think centroid is much better because it's much faster. And also from profile, maybe I need to improve a little bit more. I saw some of the like mass artifacts reported by flash deconv. Yeah.
Are there any further questions? So please unmute yourself and ask. Very welcome, everyone.
Yeah, please.
Yep. And we have questions from James, 2 questions. And I guess you can read them, right?
Yeah, sure.
Yeah. Please read. So the first one is about the issues with picking up smaller proteoforms. Could you please comment on that?
Yeah, great question. So actually, if you just look at the previous pages, like the key idea of, maybe I can go back.
Yeah, go back.
It's very long slide, so I'll try.
That was a flash presentation now.
Yeah, I tried my best, but I kind of like
yeah, that's a good slide.
Here.
Yeah, that's a good slide.
Yeah. Yes. Good. So the key idea of flash deconv started with finding the charge pattern. So that means if you have only like one single charge, basically it cannot find anything. So it was the problem of flash deconv, but when I analyzed more MS2 spectra and more native MS spectra, I found that top-down is not all about high charges, because they usually have very low charge ions as well. So I now re-implemented the recognition of masses for lowly charged ones using basically isotope patterns for the low charged ones. So I uploaded a new version, and from my test, it looked great. Not so great, but I would say it's just great. So please try the new version. And if you find more issues, then maybe I could work with you to improve the performance. Okay. And the second one is, how much time does the deep learning step add to the analysis? A very good question. So I also worried about this because the software name is Flash Suite. It should be really fast. Actually, that always bugs me. After I implement something, I need to make sure this works very fast. So it's very hard. And fortunately, this deep learning inference step is like really fast. In case of RNN, we have relatively very simple structure. So the runtime is almost negligible, less than like 1 millisecond per spectrum, or if there are so many masses, it is like 10 milliseconds, but still it is okay. So, and we are also trying to improve the speed. So I think it will not affect the total runtime that much. I hope that it answers your questions.
Well, speed is an important question, and you mentioned that in the beginning that you want to perform these deconvolution and all the procedures on the fly, right?
Yeah.
As the data being acquired.
Yes.
So based on what you presented, are we there now? I mean, is it really something that is compatible with the speed of data acquisition? And
yeah, sure. We already implemented this flash IDA thing inside of the thermal machines.
Right.
And basically this block is working inside of the machine, so it works really nice. So it is implemented, but you see here, actually, as soon as we get MS1, it processes while MS2 are acquired from the previous MS1. So we have this time, how can I say, latency. Yes. So Flash IDA performs all these steps within this MS2 acquisition and apply the selected isolation windows for the next cycle. Because the cycle is relatively short, I don't think this will hurt the performance that much.
Very good. Are there other questions?
I have a question. At the step where you are combining or you're combining data based on nominal mass, is there also a retention time component in that that combination? Because
that's very good question.
So many examples where different proteoforms would have the same nominal mass.
Yes. So for this, I didn't include retention time because the objective of this comparison was to show now deconvolution tools are making a lot of mistakes. But when I usually compare two different runs, I also consider the retention times, and I find many cases where retention times are very different but still have the same nominal masses, especially when the sample is complex. For example, even in E. coli lysase, I see many of these cases, and sometimes they have even different protein oxidation. But the other case is like because of this LC characteristics, LC in the middle part of retention time, some molecule just elute, and at the end, maybe probably because of a wash step, the same molecule elute one more time. So this is also happening, but I also saw many cases where two different molecules have exactly the same nominal masses, but with different sequences. Yeah.
Like, so one thing that I've been considering adopting, how much do those proteoforms that are identified at the beginning of a run and at the end of a run sort of influence, or do they mess up sort of the data analysis? Would it be better if the data didn't have that data in it to begin with?
Well, I didn't look into that part, but well, I wouldn't say it hurts. It does not hurt that much because even though I see some, but the percentage of such proteoform overlap is really low. But sometimes I found that, for example, like for quantification, we need to cut out the last part of LC.
Right.
So, but for identification, I didn't do that, but probably I also need to check that. Yeah, very good point. Thank you.
Yeah. You can, just not collect the last 10 minutes and not collect the first 10 minutes too, because if you don't wash the column,
exactly.
Front too, and then that messes things up.
That's a good point. But usually the last part, we do not see lots of identification because actually they are very dirty signals. So for identification, I would say it's just okay-ish, but for quantification, definitely they should be cut out. Okay, I have one question from Dave, right?
Yep. So Dave is asking a question about if we get improved MS-MS deconvolution, can we speed up the MS-MS scan rate by basically setting up fewer microscans? That's
yes.
in regard to Orbitrap, I guess.
Yes. So actually, the team, the Tolle Lab team, Philip and Conrad, we are talking about this kind of direction, like how to control these microscans, how to optimize these microscans, but we didn't reach to the conclusion yet. But I think this is totally doable, and we can control these microscans for different abundances or different range of masses. Yeah, but I don't have the correct answer right now. Yeah.
You have another MS-MS related question from Lisa. How can you filter the chimeric MS-MS spectra? And
okay,
how
great.
do you tell the chimeric?
Okay, so I also have one question from Won-Hyuk, but I will address this question.
Yeah, but that's later.
Yeah, great. Okay, so I don't like chimeric spectra. So I really hate them because, okay, about the first one. So as I just showed you, I found that many false positives are actually from this chimeric spectra. And for example, extract and also I think distiller, they report multiple MS2 spectra from the same MS2 if they have two different precursor masses deconvolved. But in most of the such cases, I found that one of them is true, the others are all false. That doesn't make sense. So maybe like for bottom-up case, we may need to come up with advanced algorithm to identify both MS2 from chimeric MS2, but I do not think we are at that stage. First thing I want to do is properly control FDR, and then if we can find how to control FDR even for chimeric MS2, then maybe we can go to the next step. But for now, I don't think just allowing multiple MS2 from the same precursor is just too risky. And about the second question, how tell they are chimeric is, excuse me, based on the MS1 full scan deconvolution, because so basically I look at the isolation window, and in the isolation window, there should be precursor masses because that's the mass I want to select. But at the same time, maybe flash deconv also reported other masses by coincidence, and some peaks are included in the isolation window. Then I calculate actually the SNR value within this isolation window, which I call precursor SNR. And I believe in the ProSite PD, it is called interference level. And in case of flash IDA, this acquisition method, it measured the precursor SNR and does not select any precursor if the precursor SNR is lower than 1.0. And the same thing goes for the flash deconv. I filter out all the precursors with lower precursor SNR lower than 1.0. Okay.
Thank you. So let's go a little bit up to the question from Won-Hyuk. And it's a very, very good question, very important. And that's, I guess, where you alluded also with the machine learning. And that's where basically you concluded that filtering of the results, the post-processing, let's say, of the deconvolution results is important. And that's the question that relates to this little overlap of results from different deconvolution software. So yes, please comment on that. How can we minimize this discrepancy?
Yeah, this is really tricky because so I try to see the actual discrepancy step by step. I started with, okay, let's go to the next page. Like I just showed you, started from different samples, different pipeline, and the same sample, and then again, like the same MS2. And actually, even though we have the same MS2, what is surprising is this. Maybe even though we have the same MS2 and same isolation windows, even though we have some discrepancy, they are both true. Because we still, like the question from Lisa, we have lots of chimeric MS2 spectra. So to reduce this discrepancy, the easy way is to just skip all the chimeric MS spectra and just select really high quality precursors. That's what I'm doing right now because that is easy, not because that is optimal. But I lose a lot, in the orbitrap case, I cannot catch up with extract or topic. I did lots of tricks, but I failed basically. So I don't know. I think the first thing we need to know is how to really control FDR in the field, in the society. We should have some kind of consensus because biologists cannot risk this kind of false positives. They want to report many novel proteoforms, but actually, even though we are saying FDR is 1%, maybe it was 30% or 50%, then what are they doing? They are just losing a lot of resources and time. So I think it would be really important for us to come up with proper FDR control. And then based on this FDR control, we should improve our algorithm. Okay, I think it can be really painful, but I believe the same step happened for the bottom-up cases. They didn't have any FDR control at beginning, but they have target decoy, works really nice. And then based on that, they competed each other and came up with nice results. So I think the first step should be to take care of this proteoform-level FDR thing, and maybe start from just harsh filtration and go to the more sophisticated precursor mass deconvolution.
And that in these lines, James is asking another question. So when you do this, what is your mass accuracy as a filter for the precursor masses? Which are the same?
Yeah, so for this, I didn't do ppm. I just calculated nominal masses, which is basically integer masses. So maybe this is not 100% correct, but I believe their tendency is correct. So it's not ppm error rate, error tolerance, but just nominal mass. Yeah. So
for smaller molecules can be a bigger thing, right? So
yeah, exactly. Yeah.
And so questionable maybe, yeah. But as you said, needs to be detailed and also looks like understanding the reasons, the discrepancy, it's something for the future, right? And as you said, maybe Dave Tapo in his coming up exciting paper will tell us more, give us more details on these discrepancies.
For sure, yeah, certainly.
or maybe Dave, you want to comment on these now? Let us know.
Goodness, that's actually a really difficult question. What I would say is that when I specify a target false discovery rate in different software tools, the false discovery rate that comes out empirically is not the same as what you target. Some of the software tools are really, really conservative and get very close to a 0% FDR when you ask them to give you a 1%. Others tend to be a little bit more expansive, perhaps through the use of machine learning. So I don't know that there's, I haven't really used the filters as intended to target particular false discovery rates, rather using a setting that seems to give a roughly comparable domain of specificity and kind of leave it at that, rather than assuming that I can recompute that. I think that Neil had a paper on computing false discovery rate, particularly for very large experiments, because if you use something like an e-value filter for prisms and say, I'm going to accept only this pile and no further, if you have 100 experiments that you all pool together, the effective false discovery rate is quite a lot higher than if you just have a dozen.
And Lisa is asking another very related question. Do you root proteoform-level FDR in protein-level FDR?
Oh, actually, what do you mean by root?
Maybe obviously it's a lot easier to have a false positive match to a proteoform than it is to have a false positive match to a particular protein sequence. So are proteoforms reported, like, do you report proteoforms that are identified at 1% FDR that are also identified at 1% FDR on the protein level?
Yeah. So basically, Yeah, you're right. So protein level FDR is no, okay, the other way around. Proteoform level FDR is more like kind of stringent than protein level FDR. So yes, if we enforce proteoform level FDR, I believe protein level FDR will be even lower. Yeah. And I think most people want to have 1% proteoform level FDR because proteoforms are the one we want to identify, right?
Good. Are there any final questions to Kyowon? Well, it's great to see that we as a society reached that stage when we started to compare the different softwares and trying to understand the differences. That shows also the maturity of the field, right? That we are reaching that as a society and looking from different angles. So that's very important work. And as this discussion has shown, it's not an easy one and there are no straight answers. And we should just continue this work going and discuss it as a community. And we look forward to do that, hopefully very soon, maybe in the next ASMS meeting. And yep, that's exactly what Lisa just said. Yes, very much so. And yeah, it's an important issue. So thank you for bringing it up. Thank you for the consortium for top-down proteomics to do that. That's there are some food for thoughts. We look forward to the future papers coming out. And we thank you once again for bringing up this topic and sharing your thoughts with us. And thank you very much to everyone, especially to Lisa and James for the many questions and today for the comments. Okay.
Yeah, thank you so much for every questions.
Everyone have a good day, and we will inform you soon about the next Proteoform Thursdays. Go top down. Everyone, bye-bye.